The whole thought was that we wanted to target the immune system using a different mechanism that hadn't really been tested, and this is selective depletion of CD20 positive B cells. MS is an autoimmune disease, right? Your immune system recognizes certain proteins in your brain and attacks them. B cells are a key part of that. So what you have is that you have, let's say, an abnormality in the function of the immune system. Taking out parts of the immune system, suppressing them, modulating, has been really the cornerstone of MS therapy for 2 decades. This is a different way of doing it because it's targeting a specific aspect of the immune system which we haven't really tested so far. These are a specific subtype of immune cells that we know are a key part of the mechanism of MS. So they indirectly or directly attack myelin, attack nerve cells, and therefore cause these disease. Now, this is a very specific subpopulation of B cells because Ocrelizumab does not deplete either the stem cells or the plasma cells. And that's important because it preserves the capacity of the immune system to reconstitute itself, and at the same time maintains the cells that make these antibodies that protect you against infections, etc. So it's very selective targeting. So OPERA 1 and 2 were phase 3 global studies, multinational studies, that involved each one of them about 800 patients with relapsing MS. So these are the people in the beginning stages of their condition. They're still having relapses. And it was a 2-year duration study where we compared Ocrelizumab versus an active comparator, a standard of care medicine called Rebif. And this is important because when we developed Ocrelizumab, we wanted to make sure that we were developing a medicine that was better than what was available now. So instead of comparing to placebo, we wanted to have an active comparator. Over the 2-year period, what the data shows is that in all major markers of MS activity, we have superiority. We're about 50% more efficacious on relapses, about 40% on clinical progression, so that's the irreversible disability that the patients have. And on MRI measures, on brain MRI measures, we're over 90% more efficacious on new active inflammatory lesions and about 80% on, let's say, scars, the less active lesions. So overall, it's really fantastic efficacy results. The safety profile in the OPERA trials is really one of the remarkable things about this drug, because over this 2-year period, essentially the safety profile is virtually identical or very, very similar to interferon beta 1A. Now, this is a medicine, the interferon, that's been around for 2 decades. People are very familiar with the side effect profile. So what you have is something that's unique in the sense that the efficacy results are very superior, but the safety is very comparable. So now you have a medicine that has really that ideal profile of a high efficacy medicine with a good safety profile. We don't think so. What you see in terms of side effects for Ocrelizumab are mainly related to the infusion. So we know with CD20 antibodies, not just Ocrelizumab, that when you're infused with them, you might get an infusion reaction, right? An allergic type reaction. And this happens in a minority of patients. So that does not happen with interferon. With interferon, the main side effects are injection as well, because interferon is a sub-Q, you have different types. So apart from those, which are different, so injection side reactions for interferon and infusion-related reactions for Ocrelizumab, the rest of the side effect profile, meaning, for example, infections, etc., are virtually the same. Ocrelizumab is given as an intravenous infusion every 6 months. So the first time you're given it, there's 2 doses separated by 2 weeks. And then after that, every 6 months, you have an infusion of 600 milligrams of Ocrelizumab.